So last lecture before the big midterm. So let's talk about the midterm and a few other things. So before I get into the midterm, I have my daily lab report tips. So after the title for your lab report, you're going to have an introduction. And I'm hoping that the introduction is relatively straightforward. The introduction is going to be about a page and a half for most people. Page minimum, 2 pages maximum kind of thing. And it's going to include some background information. So what are we talking about here? Our background, of course, our subject is membrane and beats. So you can talk a little bit about what membranes are, why they're important. You can talk about the pigment in the beats. And then you get to the point. What is the whole purpose of what we're doing in the experiment? So remember, there were 2 objectives. One was to test the effective temperatures on the beetroot membranes, and the other was, of course, the solvents. And then lastly, finish off what your hypotheses were. Again, 2 hypotheses, one regarding temperature and one regarding solvents. So that should take about a page and a half. So in your report, by the way, you are going to reference that journal paper that you found, and you can reference it. You can cite it in the introduction, or you can cite it in the discussion. It doesn't matter to me where you cite it, as long as you put it in the introduction or discussion and show me that you can do it the proper way. So after the break, I'll come back again and talk about citations. I think most people did okay on the last report. I just finished grading them, by the way, and I will get them back to you tomorrow once I'm done adding up the marks. So the methods section follows the introduction section. And the methods section, you're going to have 2 things. First, you're going to cite the lab manual. So your very first sentence should be, the detailed method for the protocol here is found in the Keanu College or Biology 107 lab manual. And there's no author for the lab manual. It's a bunch of people wrote it, but so we can just call the author Keanu College, and the year is 2021. So a sentence basically talking about where all the details of the protocol are from. And then you're going to give me not a detailed protocol, but a very, very compact, concise protocol. And you're going to try to do this in half a page. So I don't want lots of unnecessary detail. What I want is kind of just the important detail. And I'll give you an example and kind of talk about this example here in a moment. But so here's 2 examples. So you can see the first one, it says the procedure for this experiment is detailed in the Biology 107 lab manual. Okay, check, we did that. That's good. And then it goes on and talks about beet plugs and temperatures in 2 minutes. So, and then take a look at the second one. So the second one has lots of detail about rulers and razor blades and measuring and all those kind of things. So the second one is what I don't want. Okay, so the second one has all this information about, like, I don't really care if you labeled your test tubes or how you label them. Yeah, it's important that we made the beetroots in a certain way, but the construction and cutting up of the beetroots is not really as important as what the variables are and those kind of things. So you can see in the first example, it's talking about the temperature variable, right? So don't just say exposed to various temperatures. Give me specifics. So list them. There's not a lot of them. There's only 6 temperatures. So you might as well list them. How long were they exposed to those temperatures? What did you do next? So those are kind of the important things. I don't need you to tell me about how to calibrate the instrument and how to use a pipetter, all those kind of things. You're trying to keep this thing to half a page. So make sure that you're concise and that you kind of list the variables and the important parameters. The idea is somebody should be able to come along, maybe somebody, one of your peers, maybe not in this class, but another class, they should be able to read that paragraph or maybe break it into 2 or 3 paragraphs and have a decent idea of what you did. Maybe they don't know all the particulars, they don't know what instrument you used to measure this and that, but they know that you're testing what temperatures you're testing on membranes and then how you're doing to measure the membrane damage. So this takes a little bit of thinking, but like I said, you're getting it down to half a page. You're not giving all the details. You're being concise. All right, so back to the midterm. we'll come back and talk about the lab report again after the reading week. You still have lots of time to write it, and I will be available over the reading week to answer emails if anybody has any questions about it if you're working on it. So before I kind of get into the full review, I realized something I wanted to kind of talk about for a minute was the different types of eukaryotic cells. And a lot of these things we've talked about already in terms of the features and just sort of putting all in one place. So obviously animal cells are something we care about because we are animals and we have animal cells. And there's kind of your picture from the textbook showing a typical animal cell. And you can see it has all these features we've been talking about and more, some of the things we haven't talked about yet. We haven't really talked about mitochondria, for example. But you can see the nucleus, and we can see the endoplasmic reticulum and the Golgi body and all those kind of things. Often these animal cells show them having a flagellum or some cilia, and we'll talk about those after the midterm, but obviously not all our cells have that. In fact, in humans, it's just sperm cells, I believe, that have flagellum. There is an actual picture of some animal cells. So a couple of things to take note. These are things we've talked about a little bit already in some detail. Animal cells have no cell wall, but some of our cells may have an extracellular matrix. Animal cells are chemoheterotrophs. I guess we haven't necessarily talked about what that means, but that basically means we eat. We eat things to, we eat other organisms to live. So we eat other plants or other animals or fungi or whatever in order to live. There's some nice pictures, some electron micrographs there. You can see the nucleus, the nucleolus, and a lot of these structures in here are probably endoplasmic reticulum. There's another picture of an animal cell, and you can see it's loaded with mitochondria. These blue things here are ribosomes everywhere. So this one's loaded with ribosomes and mitochondria. You can see it has two nucleoluses, one there and one there. And then probably a lot of this stuff here is chromatin. You're not necessarily going to see it all. This is like a cross section, right? So what can we say about plant cells? How are they different? And this is, like I said, why I will kind of want to talk about this for a minute. You probably know that plant cells have chloroplasts, right? So not all plant cells have chloroplasts, by the way, just the green part of a plant. So that's probably the leaves and in some cases the stem. But the root cells don't have chloroplasts, and in many cases the stems are woody and are not green. But that's obviously a major characteristic of plants, that they can do photosynthesis, or at least parts of them can. Notice we have the cell wall. Remember, the cell wall is made out of cellulose. And something we talked about in the lab, of course, is that most plant cells have this central vacuole that's full of water. So there's kind of three big differences in terms of the structure of plant cells, right? The central vacuole, the chloroplasts, and the cell wall made of cellulose. There's an image of duckweed. I don't know if anyone here has a fish aquarium, but it seems that everybody who has a fish aquarium at some point gets duckweed, and it grows really well. And at first it's kind of pretty, and then after a while you get too much of it and realize you can't get rid of it. But you can see those chloroplasts. The color's been added falsely afterwards. Don't forget that plants also can have mitochondria. Okay, it's not just animals that have mitochondria. Everybody needs to make energy. So more on mitochondria and chloroplasts later. So there's just the note about what the things I talked about already. The cell wall is made of cellulose, and they're photosynthetic. So there's a nice picture of a plant cell. You can see this here. The chloroplasts, they're labeled as plastids. We'll talk about what that means later. And the color is fake, so they're not green because somebody colored them yellow. Not sure why they did that. So what about fungal cells? We did talk about fungal cell walls in particular. They have chitin. And it turns out fungal cells in some ways are more similar to animals in that they're chemoheterotrophic. That means they eat things. Now, most fungi are decomposers, meaning they're not, they don't have big stomachs and they don't chase down their food or anything like that. But they digest things. They secrete enzymes and digest things, just like us, but in a little bit of a different manner. So they have a cell wall. A lot of fungal cells have a vacuole like a plant. So they're just something different. They're a different beast. So the last group, protists, we're going to talk about after the midterm, but I just want to show you three quickly now, and we're going to see all three of these in the lab after the break. They all have unique things. So the one thing to say about protists is they're not animals, they're not plants, they're not fungi, but they are eukaryotes. They have a nucleus. In fact, if you're paramecium, take a look. They have two nuclei. They have a macronucleus and a little tiny micronucleus. And so they're kind of a little weird. So maybe a little bit more on those later. Here's another protist we're going to look at. This one here, it has chloroplasts and it has a flagellum. So it's sort of animal-like and plant-like all at the same time. So kind of weird. It's called euglena. And the other one we're going to look at is called an amoeba. There's actually a picture I took of one, and you can see they have kind of a weird shape because they actually change the shape of their cytoplasm as they move around. So much more on these guys later. We want to talk about the midterm today. So last thing I wanted to say before just really getting into the review is that topic 6, we didn't quite make it to the end. So if you're wondering what we covered, we talked about the cytoplasm and ribosomes. We talked about the endomembrane system, all the way to the Golgi body. Basically, what we did not cover was lysosomes, peroxisomes, mitochondria, and chloroplasts. So I was looking at the sample midterm yesterday, and there's definitely some questions about those things in the sample midterm and the sample questions. So if you see them, you might not know the answer, and that's why. So while you're studying, by the way, you can always contact me if you have any questions. Tomorrow we have lab 4. But that's also an office hour from me. So if you've done some studying and want to go over some of the midterm content, feel free to drop in. You don't have to just use it for lab time. If you feel it's better to use it for studying time, that's not a problem. So let's talk about this midterm here. So I'm hoping that this is not the case for everybody. Certainly feels like it sometimes, but you got to give your brain some credit. Our brains are wonderful things. And we can memorize and learn all sorts of interesting things. Sometimes it's just a matter of that recall and getting it out and doing it in a coherent way. And hopefully you're better at it than you were last semester, and we're all improving and hopefully getting smarter and wiser. So I showed you this slide. This is the plan for the midterm. So 35 marks, and you have an hour to write it. So the midterm is going to start tomorrow at 4 o'clock. And it's going to be using the lockdown browser. So I think I mentioned before what the lockdown browser is all about. And earlier today, I sent everybody a detailed email of instructions about the midterm. So it talks about the lockdown browser and what it involves. And you do have a second email I sent that says, please do the lockdown browser test. Okay, I have a 1-question test in there that will make you use the lockdown browser, and I would like everybody to do it as soon as possible to make sure that the technology works for you. I don't want to be finding out at 3:59 on Friday that somebody hasn't tried it yet and they're having technology issues. If that is you and you haven't tried it out, it is your fault if your technology doesn't work. It may affect your midterm mark. So make sure you get this sorted out. I will be taking a look tomorrow, probably somewhere around noon, to see who has not done it yet, and I will send out another reminder, but hopefully everyone will have tried it by then. So, midterm 1, we covered 7 topics basically, and so topics 1 to 6 plus topic 20, which is all about viruses and COVID-19. So I have a few slides for each of these, and that's basically what we're going to do today. If there's something in particular that people would like me to talk about in detail or go over again, please let me know. You can put comments in the Zoom chat function as we go along. But basically what I'm going to do is a little bit of an overview on these things, and I have a few kind of sample questions along the way. And so these slides, by the way, I put them up on Moodle. I think I put them up last Friday, so you can access these slides, although I have tweaked them a little bit since Friday. So we talked about studying last Friday, I think it was, so I'm just going to skip over these. And maybe the last thing I will say, for studying purposes, do check out the sample midterm and the sample multiple choice questions. And I'm hoping they will help you study. The 70 multiple choice questions, I think I mentioned before, I have taken from the textbook test bank. So they're not as carefully thought out as they were if I were to make them on my own. But I chose them all as things that we talked about or I had intended to talk about. I think there's a few about lysosomes and other things in there, unfortunately. Some of them are very hard. And so I hope you're going to go through that, use it as a study resource, and then spend some time looking up the answers and trying to understand what's going on in terms of where your gaps in knowledge are. And hopefully maybe you can contact me as well if you have any questions. I should be available most of Friday if you want to make any appointments to talk to me on Friday as well. So topic one, going way back to January. Topic one was an introduction to the cell. So we talked about the history of the cell. We talked about people like Robert Koch and Louis Pasteur and those people. And we talked about what is life and characteristics of life, and then we talked about the classification of living things and how to name them. So take a look at the nomenclature rules on binomial names, and then we finished off talking about the scientific method. And so what is a hypothesis? What is an independent variable? What is a dependent variable? And so on. Some of these things will get reinforced a little bit in the lab, but it is worth it if it's in the lecture notes to go over it and know that material. So here is a sample multiple choice question. It says, in a scientific experiment, the independent variable is, now you have to think, which one is the independent variable? So it turns out that the independent variable is the one that the scientist decides, okay? So if you think about the experiment we just did with the beets last week, temperature was one of the independent variables. The dependent variables is dependent on that other one, meaning that it's something that we measure later. So this is the correct answer for this one is A, something that is deliberately manipulated. Here's a sample true and false question. So Antony van Leeuwenhoek was the first person to observe cells using a microscope. So this was that Dutch guy, and he had those microscopes that kind of look like this, with a little handle and a little lens on it like that. So what is the answer, true or false? The answer is false. So he was not the first person to observe cells. We believe Robert Hooke was. You remember he observed cork cells. Antony van Leeuwenhoek might be the first person to have observed living cells, or at least bacteria anyway. And he did make many discoveries, just he was not the person who named cells. Topic 2 was a bit of chemistry, mostly around macromolecules. So we talked about lipids, carbohydrates, proteins, and nucleic acids. So it's important that you know what these structures look like. So I have some sample questions I'll show you in a minute. I think in the fall, by the way, I think I had what I did with this is I had a matching question. So I had 5 structures or something like that, and then words to describe them on the other side. And so you had to classify them either as a lipid or a carbohydrate or a protein or a nucleic acid. I'm not going to ask you, which nitrogenous base this is. I don't need you to memorize the 4 nitrogenous bases for this class. But you should be able to recognize something as a nitrogenous base and know that it's not, say, a carbohydrate or something like that. So we'll take a look at these sample questions in a second, and I'll get to it. So here's a sample short answer question, a definition. So just a reminder of that you will have some short answer questions, and they're all worth 1 mark, but 1 mark does not mean 1 word. It means I want basically a complete thought, which could be a sentence, it could be 2, depending on how you want to write it. So in this case, define polymer. So here's an example of a definition for a polymer. So you may remember what a polymer is. It has many something, many mers, What's the mer? The mer is a molecule, right? So this is where you have a large molecule is made out of many subunits, and there's certainly different ways to define what a polymer is. This is one definition I have. Usually when I have these definitions, I break them up into two parts. So you can see I want you to say something about it being a molecule, and I want you to say something about it being many subunits. Maybe you'll go in a slightly different direction with your definition. Maybe you'll talk about covalent bonds in between or something like that. If you're not sure if your answer is complete, give me an example, right? Examples are great, and a good example is easily worth half a mark. So, because it tells me that you know exactly what you're talking about. So do go over the definitions if you haven't done so. The vocabulary is very, very important in this class. Okay, so here is one of these matching type questions, or an identify the whatever they are kind of questions. And we can work through these and see what they are. So you can see I have sugar or carbohydrate, fatty acid, amino acid, nitrogenous base, and a steroid. Okay, so let's start with steroid. That one's easy. You look for a thing with 4 rings, and well, there it is. This is my steroid or sterol. By the way, sterol is a type of steroid. The words are used somewhat interchangeably, so sorry if I flip back and forth between the two. That one there actually is cholesterol, by the way, and I know it's cholesterol because it has a little OH on the end. So nitrogenous base, that's another potentially easy one. I'm looking for things with nitrogen in them, and actually in this group of molecules, there's only one that has nitrogen, so that makes it super easy. So here we go, we've got our nitrogenous base. And remember the nitrogenous bases, they're either going to be a 1-ring system or they're going to be a 2-ring system, kind of looking like this. All right, amino acid, okay. Actually, sorry, I was wrong. There is something else with a nitrogen in it. I just didn't see it. But it's not a nitrogenous base because it's not a ring structure. So that probably makes it my amino acid. And I know that's an amino acid because I can see these functional groups here. I can see the acid group here and the amino group. So that one's a little bit harder because sometimes when they're linked up, you don't necessarily see the functional groups in the same way. But one thing to remember is that proteins and amino acids always have that C-oxygen double bond, and they always have nitrogens in them. So not all of our macromolecules have nitrogens in them, remember? So that's one thing to keep in mind. So this is my amino acid. That amino acid is actually alanine. Okay, what about the next one? Fatty acid. Okay, so fatty acid is going to have lots of carbon and hydrogen and not much else. So that would not include this one. And that would include this one. So this is a fatty acid, for sure. So all of this is just a big hydrocarbon portion, and then this here is the acid group, the same acid group that I am seeing down here on the amino acid. And that's why it's called a fatty acid, because it's mostly a fat with a little bit of acid on the end. All right, so carbohydrate, sugars. Remember, sugars have lots of OHs. Usually there's almost 1 OH per carbon. Sometimes 1 or 2 less, but if you take a look at this one here, we've got OH, 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 OH. So that's a pretty good indicator that that is a sugar or a carbohydrate. All right, so what about the last one? I don't remember throwing that last one in there, but this last one here is glycerol. And you're probably wondering, is that a carbohydrate? Yeah, actually a lot of people would characterize it as a carbohydrate because of all those OHs on there. Some people might not because, and it's just some chemistry that I'm not going to get into, but some people would not classify it as a carbohydrate for some certain reasons around the chemistry. But I would say that's okay if you want to classify glycerol as a carbohydrate. I don't remember throwing that in there though. I think I might have another one here. So here's some other structures we can look at. Sometimes you're not necessarily seeing all the atoms. Sometimes you're seeing some colorful or representative type structures. And you can see I've used some slightly different words on here. So I've got nucleic acids, phospholipid, triacylglyceride, protein, carbohydrate. So let's take a look at these. Carbohydrate, I guess I used the word sugar in the previous slide, but remember what I said, lots of OHs, and definitely right here. So that there is actually cellulose, and cellulose is of course a carbohydrate. Or sorry, no, that's actually starch. Okay, so that was an easy one, I hope anyway. Like I said, carbohydrates, you see lots of OHs, and often you see them in these ring structures as well. That's usually another giveaway. So this one has a ring structure that looks like a carbohydrate, and in fact that is a carbohydrate, but that's part of a nucleic acid. So that's a nucleotide. Remember, a nucleotide has a phosphate, a nitrogenous base. This is the nitrogenous base, and this is the phosphate. So that there is a nucleotide. So we can confidently call this one of our nucleic acids. Okay, so anything else out there look like a nucleic acid? You're probably thinking this one here, and you would be right. That's DNA, and that's a nucleic acid. So that one is just a larger structure, so we're not necessarily seeing all the atoms individually. It's a little bit more cartoony, but that's because we're trying to represent a very large structure. So what do we have left? We have protein, triacylglyceride, phospholipid, and it turns out there's only two structures left. So what are these things? So if you take a look at the first one here, it has a phosphate group, and it's got these two long hydrocarbon tails, and so that is a phospholipid. And our last one is a protein. So all these ribbons and bands indicate that that's a protein. So remember, these ribbons and spiral structures are representing alpha helices and beta sheets. Okay, so hopefully that was decent. I'm hoping everyone has done at least a little bit of studying by now. And if you take a look at the sample questions, the one with the 70 questions, there's definitely a couple of questions there that look at some structures as well. Topic 3 was mostly on microscopy, although we did cover a few other things, such as cell culturing and cell fractionation, so make sure you know a little bit about those things as well. So another sample question, define compound microscope. So in this case here, keep in mind, anytime you have two words, make sure that you're addressing both words, right? So microscope is a device used to magnify something, and the compound part means it has two lenses, right? So you've got the ocular lens and the objective lens. So like I said, make sure you do take a look at what both words are. True or false? Oh, I gave you the answer too quickly. Some types of probe microscopy can visualize atoms, and the answer is true. So may remember that little video I showed you from IBM where they were moving around individual atoms to make that little boy bounce the little ball. And so you can definitely visualize individual atoms, which is just really remarkable. So you are going to have a couple of long answer questions, and by long answers, I mean they're worth 5 marks. So if this was a written exam with a pen, that would be about somewhere around maybe 2/3 of a page to a full page of written material. Typing, it might be a little shorter, but so 5 marks. If you think about what I was saying with the 1 mark question, now I'm thinking about 5 complete thoughts. So some of my long answer questions are like this, and I think I mentioned this one before, this exact question. You can answer it in a table, and so, you know, you want at least 5 points, okay? So if you think about comparing some of the items, right? So if we are comparing a light microscope and an electron microscope, what is the light and electron part? So that's the illumination source. So you can talk about that. And if you want to be extra thorough, you can tell me that different types of light sources can be used. So we could use a light bulb, we could use the sun, we could use a laser. Electron microscopes, there are different types of electron guns, but those are things that we've talked about. And then you want to talk about other variables, right? You can think of some of the techniques we mentioned. fluorescence and staining and so on. You can talk about how on a light microscope you can actually look at living specimens. In an electron microscope, they're dead. And so there's actually a lot of things you could discuss with answering this particular kind of question. But do make sure you address some of the key things. So you can see here, for example, it says, give an example of something you might see under the 3 types. So I can't remember whether I had this question in the fall, but I do ask this particular one. I've asked a few times in the past few years. And I've noticed sometimes people give me a great answer and then they don't give me examples. Or they give me examples of light and electron samples, but they don't address both SEM and TEM. So do make sure you're thorough on this kind of thing. Like I said, 5 marks is worth at least 5 points. If you're not sure if you have enough material, give me a couple of more, okay? Because, you know, I mean, if you're not very detailed in your answers, like if you just say, you know, electron microscopes can see smaller things, well, that's not a lot of information. I'm not going to give you a full mark for that. But if you can tell me the exact magnification or something like that, then those are better details to give me. So you do not have to answer exam questions in full sentences, by the way. You can answer in point form. I don't mind on exams. It's the assignments that I do want to see proper sentences. But you know, like I said, answering in a table makes it easier to grade, and it's easier to follow and organize, and that's always good for these kind of things. Topic 4, we were talking about membranes, membrane transport, and cell walls. So regarding membranes, make sure you know what each of these things are and what they are doing in the membranes. Okay, I think last semester I had a question, was it last winter? I had a short answer question and I said, what are carbohydrates doing in cell walls, or in membranes? And half the class didn't know the answer to that. That was kind of key. That was something we talked at the beginning. The 3 components of membranes are, and people should know what those carbohydrates are doing. And if you don't know what they are doing, go look that up. I'm not going to tell you right now. So here's a sample multiple choice question, and it's asking what cannot easily get past a plasma membrane. So remember that phospholipid bilayer is hydrophobic. At least the lipid part is hydrophobic. So hydrophobic things can cross easily. Uncharged things, usually. Non-polar molecules, that just really means hydrophobic. Small things, if they're really small, they're good, like water. Charged things, the answer is no. Those cannot cross the membrane very easily. And so that is the correct answer. The exception kind of with polar things is water, because water is super tiny and is really, really abundant. So it's kind of hard to keep all the water back. It just eventually some sneaks through. So let's talk about these 3 things and you know, what are they, right? Because they're all different things. So remember that cell membranes are made out of phospholipids. And we just mentioned the other 2 things. Hopefully you remember what they are: proteins and sometimes carbohydrates. So what kind of cells have these structures, cell membranes? All cells. All cells do. And what's the function of a membrane? That's kind of like, that's a whole essay, what a function of a membrane is. But it separates a cell from its surroundings, separates cell from surroundings. So sorry, just hard to squeeze everything in there. And the other major thing is it allows transport of stuff inside and outside of the cell. So I'm just going to put the word transport. So what about a cell wall? Remember, it depends on who you are, right? If you're a plant, your cell wall is made of cellulose. If you're a fungi, your cell wall is made out of chitin. And if you're bacteria, your cell wall is made out of everyone's favorite word, peptidoglycan. See if you can say that nice and smoothly. It runs off your tongue just beautifully, doesn't it? We did talk about other cell walls, but those were the 3 that I said, no, these ones, okay? What types of cells have these structures? I guess I just answered that. Maybe I can say not animal cells. So what is a function of a cell wall? We're talking about this gives a cell shape and protection. So very different than a membrane. Cell walls are leaky. Molecules can move through them very easily. It's not about transport. It's not about separating solutes or anything like that. It's about protection against osmotic pressure and protection against exterior forces as well. So what about extracellular matrix? So the composition, it kind of depends on what kind of extracellular matrix you're talking about. But there were two that we talked about in class. We talked about collagen, and that's found in skin and connective tissue. And we talked about calcium minerals, and that's found in bone. So both of these, of course, are animal cells. The function of an extracellular matrix, it depends on the cell type. I'm not gonna write an answer there, you know, bone is obviously strength and protection and allows animals to be mobile. Connective tissue, you know, because we're mobile organisms, it's really important we have our kind of elasticy connective tissue so that when we move around, our tissues are able to do so. And but I'm not going to get into that in any more detail than that for this course. All right, moving on. I hope everybody got that, and please excuse me for my handwriting. It's really hard to write on computer screens with any amount of neatness. It's just not easy at all. Topic 5 were the prokaryotes. Remember, the prokaryotes included both the archaea and the bacteria. So let's see if we can remember what these parts are and what they do. So number 1 are these hair-like structures, and these are called fimbriae, and the other word is pili. And actually, 1 L or 2? Why can't I remember that? I don't know. I used to be able to spell a long time ago, apparently, just not this year or last year, maybe the year before. Okay, number 2 are, it is pointing to that gel inside the cell. Or no, it's not. Actually, we already have the cytoplasm. So 2 is a ribosome. Just, okay, somebody who did the arrow. Not me, I didn't do that arrow. Just a bad arrow, not showing the ribosome. Ribosomes are those little specks in there. So those are involved in protein synthesis, of course. I don't know if I mentioned what these are. These are sticky things. They are involved in cells attaching to each other, unless it's a sex pilus, which is involved in bacterial conjugation. So number 3 is not the nucleus, but is the nucleoid. So remember that a nucleus has a membrane around it, the nucleoid does not, but it's still, the DNA has to be somewhere. So somebody gave it a name and said, this is the nucleoid region of the cell. Meaning that this is where all the DNA is packaged up. An inclusion, that just means it's a piece of food. So 4 is a ring, and it's a ring of DNA, and that is a plasmid. So it's kind of like a mini little chromosome with a few extra genes on it. Sometimes they have antibiotic resistance, and that's something that we're concerned about when we're following new infectious organisms. Number 5 is the tail, and the tail, of course, is the flagellum. If you have many, you have flagella. If you have 1, it's a flagellum, and that's used for swimming. So 6, 7, and 8 are the cell surface, and so 6 is the plasma membrane. So there's our membrane there. 7 is the cell wall. Remember, it's made out of peptidoglycan. And 8 is outside the cell wall, and not all cells have it, but there are capsules, and the other version is something called a slime layer. Remember, slime layers are just generally a little bit more sticky and goopy than capsules, but you know, if you discover the organism, you get to decide whether it's a capsule or a slime layer, really is the way it is in the end. And both of these things are called glycocalyx, and that means sugar coat. And they're used for sticking to surfaces and also escaping immune cells. I'm just looking at my archaea up here. I'm just, my writing is bad. I apologize again. I can't seem to figure out how to erase using this pen. Sorry about that. Some sample questions. Again, definitions. What is amphipathic? So remember that that is a molecule that has both hydrophobic and hydrophilic components. And if you're looking for an example is a phospholipid. What is an endospore? So an endospore is a thick, coated, dormant structure, and it's produced by some types of bacteria when they are suffering some environmental conditions that are harsh. So it could be lack of water, lack of food, or something like that. So like I said, notice these are kind of complete sentences or complete thoughts at least, and they have multiple parts to it. And that'll be part of my answer scheme when I have these short answer questions in that, I'm not looking for one word. So be thorough, give examples. Topic 6 was eukaryotic organelles and eukaryotic organisms. So let's take a look at what we have. Number 1 is the nucleus. So hopefully that one is super obvious, and that is where the DNA is. Also inside is the nucleolus, and that is where the ribosomes are being put together. Number 3 is the Golgi body, or the Golgi apparatus, or the Golgi complex. I don't care which of those you use, just get the Golgi part in there. And that is where proteins that are made are shipped to, and then they get distributed to other parts of the cell. Number 4, of course, is the endoplasmic reticulum. Remember, there's the rough and the smooth. The rough makes proteins, and the smooth makes lipids. Number 5, mitochondria. I won't ask you about mitochondria. We haven't talked about them really yet. Number 6 is the plasma membrane. Number 7, peroxisomes. I haven't asked you about those either, so don't worry about those. We have the cytoplasm and lysosomes, so I'm not going to ask you about lysosomes either. So make sure you know what these organelles do. And I usually have, I think the last few years I've had some sort of matching question around organelles or cell components. Sometimes I make it thematic. Sometimes the question may be all eukaryotic components, or sometimes I have a little bit of both. But usually I'll throw a matching question of some sort in there around some of these things. So let's just review a few of these things. Sometimes my short answer questions are fill in the blank style. And so it'll be like, maybe 2 blanks is 1 mark or something like that, depending on what I decide. So this could be a type of short answer question. What organelles do each of these functions? Translation of membrane proteins. Wonder if I even have the answer in there. No, I don't. I'll have to write it with my pen. So translation, you got to remember what that is, of course, right? That means protein synthesis. But this is of membrane proteins. So what are we talking about here? We are talking about the rough ER, the rough endoplasmic reticulum. That is where membrane proteins are made. Cytoplasmic proteins are made by ribosomes in the cytoplasm. Membrane proteins are made by ribosomes in the rough ER. Number 2, synthesize cytoplasmic proteins. Okay, so this would be cytosolic ribosomes. So if you just, wrote ribosomes, sometimes they give you the full mark, sometimes a part mark. So, give me what you have, right? I know not everyone's going to know everything. Ribosomal subunit assembly. So that is in the nucleus in a special place called the nucleolus. Number 4, support the nuclear membrane. Okay, so we just talked about this last day. I know it's fresh in everyone's head, but maybe you don't remember what it is, which is why you have to study. So this is called the nuclear lamina. Remember, these are protein fibers, and they are just inside the nuclear membrane, and they're giving it shape and structure. Number 5, glycosylate proteins. So this means add a glyco unit, which is a carbohydrate, and this happens in the rough ER, and it happens in the Golgi. A little bit happens in each. Depends on the protein how much happens in each of those organelles. Number 6, break down old organelles. We haven't talked about that one yet, but I'll give you the answer anyway. That's a lysosome. So lysosome is kind of like a digestive compartment, kind of like a cellular stomach. Number 7, photosynthesis. We haven't talked about that yet, but I'm hoping everybody knows the answer to that, which is chloroplasts. Yay, photosynthesis. We use a little bit of that and some sun right now. So here's another sample long answer question. I think last semester I gave a similar one. I think actually last semester I asked everyone to compare bacteria, archaea, and eukaryotes. And so just re-emphasizing what I had said before, you better give me at least 5 points. So, and of course you have to tell me which of them have a nucleus and which of them don't, and you have to have that information correct. Just disregard this part. Sometimes I've given 3 questions and you answer 2, and I don't really know how to do that in Moodle. So I'll just give you the 2 questions that you're going to have to answer, and try to mark them a little bit more generously than usual. So one more unit. Don't forget that we talked about viruses, and we talked a lot mostly about SARS-CoV-2. So remember that SARS-CoV-2, that's the name of the virus. That means severe acute respiratory syndrome coronavirus 2. And the disease is COVID-19. So that means coronavirus disease 2019. The D is for disease. So don't forget which is the virus and which is the disease. A lot of people get that one mixed up. So I have a couple of sample questions about viruses. So what type of genome does SARS-CoV-2 have? So that was just on the previous slide. So maybe you remember, maybe you don't. It's an RNA virus, and it's a single-stranded RNA virus. There we go. Viruses, to my knowledge, never have both. We also talked about vaccines, and so this is talking about a particular type of vaccine that is produced using a weakened version of the pathogen. What kind of vaccine is this? So it's definitely not just messenger RNA. It's definitely not just a protein, because those are just parts of the virus. So it's going to be one of these ones here. And hopefully you remember that the word we used for a weakened version of a virus or another pathogen is attenuated, and that means weakened. All right, couple more things to wrap up today. Here are the major concepts to study, and I guess I should have put on there viruses, virus structure, or SARS-CoV-2. So, you know, this here is a major theme of the course. You know, make sure you pay attention what, you know, what's going on with these structures, where are they found, who does it, what kind of cells. So that's kind of important for that. Scientific method, macromolecules, types of microscopy. So there's all going to be a few marks on each of these things, which is why I'm telling you to study them. Make sure you look at the sample midterm, and make sure you look at those 70 multiple choice questions that I put on there. Like I said, some of them are pretty hard, but I'm hoping it will help you with your studying. Lastly, I have a little good luck balloon for you. And so I'd love to give you that in person, but this is the best I can do for now.